I will briefly explain my work titled as Mechanistic Studies on Ubiquitinin Membrane Interaction and Development of Infection Imaging Probes. Due to prevalence of focal infectious diseases such as fever of unknown origin, osteomyelitis, soft tissue infection, etc., there is a need to develop non-invasive methods to detect infections at an early stage and distinguish them from sterile inflammation to achieve better treatment outcome. So far, only limited success is achieved in development of radiopharmaceuticals for detection of infections using non-invasive methods like SPECT and PET. The objective of present work, therefore, was to develop such infection imaging probes. Molecules like antimicrobial peptides are useful candidates for this purpose as they can potentially distinguish between mammalian and bacterial cells. In this study, two peptide fragments UBI 2941 and 3138 derived from a longer antimicrobial peptide ubiquitinin were utilized for this purpose. To study interaction of these peptides with membranes, model membranes mimicking either bacterial cells or mammalian cells were prepared. It was observed that UBI-derived peptides could only bind to the bacterial membranes and not to the mammalian ones. These peptides were also taken up by live bacterial cells. Furthermore, interaction between membrane models and UBI peptides was studied using several biophysical techniques. It could be concluded that UBI peptides selectively associated themselves with bacterial membranes due to electrostatic interaction, and they underwent conformational change in this process. This membrane peptide association resulted in decrease in membrane fluidity. UBI 2941 had a more drastic impact on membranes as compared to UBI 3138. In order to develop infection imaging probes, the peptides were labeled with 68 gallium, which is a positron emitting radioisotope and hence facilitates imaging of tracer by positron emission tomography as well as luminescence imaging due to Cherenkov effect. 68 gallium labeled peptides were specifically taken up by bacterial cells. Luminescence imaging in mice model of infection showed that UBI peptides accumulated only at infection foci and not at the sites of sterile inflammation. 68 gallium UBI 3138 could detect infection with higher sensitivity. Under limited clinical trials in human patients, 68 gallium UBI 3138 could successfully detect lung infection in one patient and soft tissue infection in the thigh of another. To conclude, through this research work, UBI membrane interaction was successfully utilized for development of positron emission tomography based infection imaging agents. Thank you.